Good afternoon. This is Dr. Dan Guerra, and you're watching and listening to Authentic Biochemistry, the podcast that's all about, obviously, not just biochemistry, but the authenticity of the science, particularly the research science that we discuss. Been a while since I covered a video lecture. I normally do audio lectures, and my podcast can be found on all the normal platforms. Just look up Authentic Biochemistry, and you will discover it. And that would be me. My name is Dr. Daniel J. Guerra. Now, today, we are going to be doing what I'm calling an interim lecture in a long series that I've been doing for a couple of months now on immunoepigenetics. These interim lectures are so that we can fill in some major gaps in most people's knowledge base concerning the fine points of whatever aspect I need to cover. Today, we're going to be doing a discussion of nucleic acid torsion angles. Hopefully, that is your cup of tea, or in my case, cup of coffee. Actually, cold espresso coffee. So let's just get started. If you haven't watched my podcast in the past, it's your first time, you are really in for a treat. If you've watched them before, then you already know you're in for a treat. So let's do this, okay? 11th of June, 2023, that's the authentic date. And again, we're going to be talking about thermodynamics, particularly about how thermodynamics of DNA binding proteins that are associated with methylation of nucleic acids in an epigenetic pathway can be discovered and understood via thermodynamic considerations. Okay, so let's just go. So this is from a paper you can see, Nature Structural Molecular Biology, published some 5 years ago, actually. What is this paper going to reveal to us? So as it written there, cytosine bases in CpG islands, or sites, in genomic DNA can be methylated, of course, as we've been saying for a very long time on this podcast, by methyltransferases such as DNMT1, or 2 or 3, the different isoforms, which are most canonical in humans. They will produce a product called 5-methylcytosine, or MC, and you're going to get demethylation of that methylcytosine, and that can involve a sequential oxidation, first to 5-hydroxymethylcytosine, then to 5-formylcytosine, which is going to be a major feature of today's talk, and then the complete oxidation to 5-carboxycytosine. Now, the enzyme that carries this out is the 1011 translocation dioxygenase, known as the TET. The oxidized pyrimidines, such as formylcytosine and carboxycytosine, can be excised directly from the deoxyribose sugar by an enzyme called thymine DNA glycosylase. Thus, subsequently, it can be replaced by an unmodified cytosine. So you've removed the modified base. That is a part of the pathway known as BER, or base excision repair. So DNA has multiple repair pathways. Some of them involve recombination mechanisms and not homologous end joining. This particular one is more about just removing specific atoms or structures of the molecule within the DNA helix to then form the substrate to be able to add back the authentic nucleotide in that position. Okay, that's what base excision repair is, perfectly named. Now, formylcytosine, which is the oxidation product via that dioxygenase, that TET dioxygenase, is a substrate for TDG, the enzyme that will remove that modified base. But because formyl groups have an aldehyde functional group, and because of the mechanism of the enzyme, it's potentially going to be a remaining formulation, thus a maintenance formulation in the nucleic acid. We talk about maintenance methylation for epigenetic signatures. This could be maintenance formulation. Okay, so you understand the nomenclature. So if you look at this structure here, you can see the DNA 5-methylcytosine methylated site in the 5th position of cytosine on that carbon. Okay, it's right there. And you can also see that you can methylate adenine, or when it's bound to the sugar, adenosine. So there is a 6-methyladenosine modification that will occur in the 6th position of that nucleotide. That can occur in DNA or messenger RNA or in microRNA. Also, I should point out that it occurs in ribosomal RNA and in transfer RNA. So all the RNA molecules. Now, the writers for epigenetic, the authors of epigenetic reprogramming of gene expression, because that's what epigenetic modification will do, alter the expression of the genes that have been epigenetically modified, such as with this methylation pattern. They include enzymes like DNMTs and also the N6-AMT1, so that you can see the products of those reactions, right? Now, the readers, once you author, you author a methylation pattern, then that methylation pattern has to be read by other polypeptides. So these readers also can modulate the script that has been added. But typically all of them, if they're working on a methyl group, will have a domain in the protein called an MBD, simply a methyl binding domain. Okay. Now, erasers of that methylation can be these 10-11 proteins that we've already introduced to you, or there can actually be other oxygenases and sequence-specific demethylases. Okay? So we talked about these proline-associated oxygenases in the past. We talked about the mechanism of the TETs. We also spent a lot of time talking about demethylases. So remember, this is a lecture deep into the series. So I'm not going to go through all that now. Just if you first tuned into my podcast, just understand we've covered all of these. Right? So remember what epigenetic modification does. It alters the expression of a gene without directly changing the nucleotide sequence. It modifies the nucleotides, but doesn't change the sequence. Okay? And this can occur on mitochondrial genomic DNA, as well as nuclear genomic DNA, okay, in a mammalian cell. And in plants, also the chloroplastic DNA can also be modified. These modifications then, because they alter gene expression, will alter cell fate, as well as modifications of simply just gene expression, going from, say, a pro-inflammatory T cell to an anti-inflammatory T cell. It's one good example. So what are the mechanisms? DNA methylation, again, you already saw the products. They require unique enzymology. The 5-methylcytosine is the result of methyltransferases. And you can see here, there are the 3. DNMT1 is the primary maintenance methylation author. And that status of methylation can exist across cell division. While the other 2 methyltransferases, 3A, 3B, work in a de novo methylation process. So what I mean by that, you have a maintenance methylation at certain genomic loci that can be carried over between cell divisions. And then often you also have a methylation-specific modification that does not survive cell cycle. If that's going to occur, these authors, DNMT3A and 3B, tend to be more often utilized as the enzymes that carry out the reaction. We already went through this whole discussion here about the TET. In fact, there are 3 different TET proteins. You can see here, again, this is a passive dilution. For example, when you take a cytosine, methyltransferase, you make the 5-methyl-C, and you then start to oxidize by making 5-hydroxymethylcytosine. That product can be passively depleted through DNA replication. So it's lost. That authorship is lost. Again, if you follow through the oxidation, you're going to then make this formyl, and obviously at the end, this carboxy derivative. Okay. Now, each of those can also be modified through base excision repair. Okay, so there's 5-hydroxymethylcytosine. This enzyme AID, which is adenosine deaminase, will make 5-hydroxymethyluracil. Then that TDG enzyme, remember thymine DNA glycosylase, will then repair that entire locus and bring back the cytosine residue. And that can occur with the hydroxy, the formyl, and even the carboxy. Same enzyme. Okay. Now, let's go into more detail about this. When you have formyl cytosine and it's present in CPG repeats, also known as islands, that are linked to promoters or enhancer regions that are going to be 5' and proximal to the open reading frame for transcription of any given gene. Besides affecting the transcription rate of the gene in variable ways, which we've talked about and we will talk about again soon, what happens actually to the physical chemistry of the DNA? Well, it causes a perturbation to the DNA double helix. That can result in a unique helical conformation, which will provide for a 13 base pair turn in the DNA. Now, this formyl DNA might be the basis for epigenetic recognition of formylated CPG islands in DNA, obviously, right? Because you're having more of an effect not just on that one cytosine residue, but on residues proximal and distal to that formulation. Okay, it's an important feature of today's lecture. Now, when you look at the crystalline state, you make a crystal out of these reaction products. Using X-ray crystallography, then, followed up with that, you get the equivalent unmodified duplex and 3 sequence variants. Now, they seem to have almost identical structure to DNA containing multiple 5-formyl cytosine bases. And thus the structure could be a member of the A family of DNA. So all of these names, A, B family, etc., are well described in the literature, conformational structures of DNA found in the nucleus. Okay? Now, using NMR, nuclear magnetic resonance, of that formyl cytosine DNA, and then the native analog, you can see that actually what's occurring is you generate B family helices. And that occurs in solution, not in the crystal. Okay, so the difference, of course, is going to occur when the DNA is in solution, because you have solvation effects or solvent effects. So despite the circular dichroism spectra of the FC-containing duplexes displaying unusual characteristics, it looks like you really have a B configuration. So the paradox here, or paradox is thesis, antithesis, no synthesis, is a consequence of local electronic transition dipole moment changes. which we covered in a few lectures back. That's caused by formyl cytosine sensu stricto, rather than gross structural perturbation of the entire DNA macromolecular double helix. Okay? So what is this showing you here? Why is there a paradox? Why is there a discontinuity in the evidence? Because when you look at an X-ray crystal of a protein bound to DNA, it is already a dead system. And so you isolate it as a crystal. When you're looking at that same structure, product of the reaction, right, the methylation, and then the partial oxidation to a formyl group, but you're looking at it in solution, and using NMR, you get different data. So this is another important issue in understanding research science. The method you use to study, for example, a biochemical system, like what I do, what biochemists do, can have a role in the evidence that's generated, the data that's generated. And you must know how your instrumentation can modify the evidence. You must know this a priori. Okay? Otherwise, you'll publish papers and say, well, we know that you get this A structure from formylated cytosine. But wait a minute, that's in the X-ray, that's in the crystal. What happens when you look at it in solution? Well, you just have a different P family helix. See, and that plays a role in the thermodynamics of DNA metabolism relative to recombination, repair, replication, and transcription. Yeah. So you have to be careful what kind of induction you do after you do your research. Now let's get some more intense detail. There are torsion parameters, torsion angles in DNA. And those are actually very important in an empirical force field generated for that particular biomolecular system that is of chromatin in the nucleus. Now, because these torsion angles affect conformational equilibria, remember, you can have different kinds of conformers once you make the modification with a methyl group. And since the modest changes have significant influence on the population of nucleic acid in general, they are relevant to all the processes involving those conformational changes. What are those processes? I just went through them. Recombination, repair, replication, and of course transcription. Okay. So nucleic acids have these conformational states and they have biological significance. That's the important point to make about that. Now, the equilibrium between two different B states, the B1, B2, which are called substates of the B family of DNA conformation. You can also get AB or BZ forms. Right? So you can have BDNA, zDNA, or you can have equilibrium conformer manifestations in the nucleus. And these all are going to modify those four processes I told you about. Okay? So that means you're going to be looking at the properties of the DNA duplex, sensu stricto, and all of the non-canonical RNA motifs, which also can be obtained by epigenetic modification. So that means accurate modeling, absolutely accurate modeling, if you're going to be doing modeling in silico of torsion potentials, will be a significant problem when you're doing simulations. Okay? Now, doing an X-ray crystal, that's not a simulation. That's laboratory research. Likewise, doing solution chemistry of DNA-bound proteins. That is bench level, wet biochemistry, right? That is test tubes and centrifuges and purification columns, all of this technique, right? It's not in silico. Now, you remember what the in silico means. That means it's all done with a computer. Modeling is all done with a computer. Okay, that is informed by evidence from the research literature. But once you're doing the modeling itself, and then that's the product of the study, then it's completely in silico. That means it's a biomolecular simulation. Got it? Okay. So torsion parameters are most frequently studied components. They are the things that most often people want to study when they look at nucleic acids as polymers. Any minor inaccuracy can have a major effect on the population of different structural forms, obviously, because there's a great deal of DNA in the nucleus, right? A lot of mass is covered there. And that will lead to artifactual geometries and incorrect estimates of free energy coefficients between the different conformational states. And all that's significant if you're a physical biochemist and you want to know all of those discrete geometries and free energy potentials of the nucleic acid you're studying, because that's going to play a role in the biological activity. Okay. So trajectory instabilities have been detected in simulations of DNA and RNA. And that's because of the long time scales that are necessary to watch the event play out. But these are events, these aren't substances. Everything is always an event ontology. You've heard me say this 100 times. Right? So when you have something that's actually occurring in time, those slight instabilities, those slight inaccuracies will give you huge inaccuracies in the actual torsion angle distribution, which can make your data no longer evidential. Right? So evidence-based refinement of the torsion potentials of authentic nucleic acid force fields always are going to require further analysis. Evidence-based means actual research at the lab bench of DNA protein complexes and not modeling. Okay. So here are the different torsion angles we're talking about in DNA. This B1, B2 is not the bDNA, those base 1, base 2, running 5', 3'. So you have here beta, these are torsion angles, gamma, delta, you notice where they are relative to the carbon atoms. There's the epsilon torsion angle. Then around the phosphate, this is the sugar phosphate backbone, you have the zeta torsion angle. There's the alpha torsion angle. There again is your beta gamma, like up there. The delta, the epsilon, and the zeta. You see how it follows each base pair. So when you add the base pairs together, they're stacked together. When you do that and look at the torsion angles of a nucleotide that's proximal to one below, which would be distal to those torsion angles, we talk about steps, steps in that concantimer DNA strand, right? And those steps of torsion angle that you measure are going to be altered 5', 3', or even 3', 5' based on any modification you did, covalent or otherwise, to the nucleic acid at the sugar level or at the pyrimidine or purine base level. Okay. So, optimization of torsion potentials involves fitting the torsion energy from a computer-generated calculation. So the average torsional angular twist is often underestimated. You can understand those torsion angles are going to be involved in twisting that DNA around those axes. It's going to be underestimated compared to experimental evidence. That's not trivial since the errors in the twist are cumulative with an increasing sequence length, obviously, and underwound duplexes, thus adding significant error in modeling the geometry of the nucleosomal or mitochondrial, for example, circular DNA. So torsions in nucleic acid backbone, like the epsilon torsion angle, is defined as the C4 prime, C3 prime, these are carbon atoms, oxygen 3 prime. Phosphate and zeta is defined as the C3 prime, oxygen 3 prime, there's the phosphate, oxygen 5 prime. Okay, so that's where those angles are found. Right? All right. So you've got the epsilon and you've got the zeta. Now, just to remind you, why are those numbers given primes? Because nucleic acid nomenclature, which is based on biochemistry, the atoms that are in the nucleotide sense restrictor, that is the adenine, the guanine, the cytosine, the thymine, or the uracil and RNA, right? Those carbon atoms are not listed as prime. The carbon atoms in the sugar, the ribose sugar, are given the prime. So you can differentiate those when you're talking about the chemistry of the nucleic acid. So you number these ribose sugars from the carbon to the right of the oxygen atom, right? It's the way it's formally done in these configurations. So it's 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. That relates directly to what I just showed you, where these torsion angles are. See, that's the primes. See, that means it's on the sugar. You don't have to worry about showing a prime for the phosphate. Why? Well, because you're only talking about the phosphate and the sugar, whereas these carbons could be on the nucleotide or on the sugar. See? All right. So it's all very logical. All right. Now, back to these studies. Two highly correlated epsilon zeta torsion angle states commonly found in DNA. The canonical dominant, which is also known as the trans GB1, and a minor but significant G trans B2 state. Okay? So it's different names. We're talking about those two torsion angles I just showed you, where they are specifically on the backbone, sugar phosphate backbone. Okay? So B1, B2 equilibrium deserves accurate study because it involves nucleic acid recognition by protein. Obviously, the protein is going to bind to a specific region of DNA. We need to know if it's going to have an effect on torsion angle chemistry and physics. So B2 will exhibit more uniform distribution of backbone angles and all of the helical parameters. And they have negative rho, increased twist, and sugar pucker, where the sugar is puckered out of the strand of DNA. Okay, it's close to what's called the north configuration, right? Or I guess we could call that adaxial conformation. So the B1, B2 relative populations are sometimes not modeled correctly. Why are they not modeled correctly? Because there's too much authentic biochemistry going on for the modeling to deal with these alterations. Because these are not alterations that are fixed in every B1 or B2 conformer. Okay? And because they're not fixed, right? You have to measure it. You can't simply map it onto a computer program. You have to measure it. So these are where all these errors are in this molecular modeling that goes on. Okay? So some of the reasons B2 states can be found at any position in the sugar phosphate backbone, but their occurrence depends on a base step function. That means one base on top of another. Okay? Think about where those torsion angles were. Some steps are conducive to the B2 state. So this is what it would look like. So Y is pyrimidine and R is purine. So pyrimidine, phosphate, purine. So and the GPC is where Y stands for pyrimidine and R stands for purine. So in this case, the C is the pyrimidine, excuse me, and G is the purine. You see how that works? Okay. So this is an actual nucleotide, guanine, right? And that's a purine. And what is cytosine? It's a pyrimidine. So you can see how the nomenclature puts together. You also remember the DNA has counterions, sodium versus potassium, and that plays a role on the modulation of the population of B1 versus B2. Once again, solution chemistry versus crystals, and certainly versus molecular modeling. Okay. So the occurrence of the B2 stage in one strand increases a probability of finding another B2 state opposite that given base step, because you have two strands, right? That can promote a B2B2 step in terms of torsion angle conformation. Now, in contrast, the presence of a B2 state lowers the probability of finding another B2 state in neighboring steps. Remember, the nucleic acid isn't stretched out like a ladder like I showed it there. Nucleic acid is compacted into the nucleus and held together by nucleosomes. And nucleosomes have an octet of histones where the DNA is wrapped around, right? And so that compaction of the DNA is necessary to have all of that DNA in a very small, relatively small part of the cell, the nucleus, an eukaryotic cell. Okay? You have to keep all of that in mind when you want to be accurate. And what we have to be is accurate. We're discussing how epigenetic modification has an effect on the thermodynamics of DNA metabolism. You see? Now, if you use P31 NMR, nuclear magnetic resonance, that particular method of analysis, you're going to rely on empirical correlation between the chemical shift of the phosphorus, that's going to be the delta P, right? It's a change, a delta P is a shift in phosphorus, correlated to the percent of B2, assuming that the delta P in the B1 and B2 states are sequence independent. And that delta P depends only on which population of conformer you're talking about. Okay, that's how you make that assumption when you use P31 NMR. Sequence dependence of that delta P can arise when base pair steps differ. However, in their helical geometry and an average value of these torsion angles, alpha, epsilon, and zeta. Okay? That bit of evidence suggests that sequence effects may significantly alter B1, B2 populations calculated from the measured P31 NMR. Okay. This has to all be part of the process of examining the data. And for that matter, designing the experiments. Now, the study reports that a reparametrization of the epsilon and zeta torsion parameters or angles rely on calculations and accounts for the conformation-dependent solvation effects. Okay. Remember, there's a solvation effect, solvent effect. We talked about this a couple lectures ago, how even when you have a methyl group, part of the hydrophobic interaction between the methyl group and the methyl binding protein from the protein that, for example, is an author or reader of that methylation on that cytosine residue, it's going to involve an area of hydration of the DNA, right? Because the hydrophobic effect, you're going to be concentrating the hydrogen bonding, therefore enriching certain centers around the nucleic acid in water, and the other areas are going to be deficient in that water. And that's how the hydrophobic effect works. That's why it's so strong, particularly in macromolecules like proteins and DNA, and of course lipids. Now, Continue on. The main differences with respect to the original parameters were that when these studies are all analyzed, stabilization of what's called the flipped epsilon zeta torsion angle state. So they can go from this kind of status to that kind of status. They can flip. Okay? That's what torsion angles can do, as long as they are allowed to, based on the steric hindrance of the sugar phosphate backbone. Okay? There are allowed and non-allowed regions of reorganization, or they're calling it reparameterization. Anyways, that occurs, and that lowers the transition barrier. Once you get this stabilization of the flip state, it's going to lower the barrier between going from B1 and B2, obviously. So it is a magnification of the effect. Once you start enhancing this alteration of torsion angles, you can then enhance the reorganization or reparameterization of subsequent torsion angles. You see? That's the way it works. So you can call it a snowball effect if you want to. That's another way of looking at it. So this epsilon zeta OL1 correction. Now, I didn't get into what that is. That OL just stands for, that's an acronym for the city in which the researchers were working. Okay. It doesn't have any meaning in terms of, those aren't chemical signatures. Okay. So we'll just call delta E correction on average increased populations of the B2 substrates and increased B1, B2 interconversion rate in the bDNA duplexes. Okay. So there was a correction added because of the solvation effects. And that was necessary to be able to look at all the subsequent data generated. So whereas most bDNA structural parameters remain unaffected, this correction, okay, of those two torsion angles, several desirable effects became apparent. This correction increased the average twist in this simulated bDNA duplex, mainly in the base pair steps where it was previously significantly underestimated. So see, these researchers are taking the biochemical data from solution chemistry of the DNA bound to the protein, and then adding that to their modeling. And that's what that correction is. That delta zeta OL1 is a correction to deal with the solvent effects, which are relative to the stacking or the steps of the individual bases, relative to the position of those torsion angles. So the resulting twist and sequence dependence are better agreement then with the solution NMR data with the correction. Okay? Besides that, this correction improved the description of groove DNA as a major and a minor groove. It gave a better coupling of the actual authentic measurement of those groove widths. So therefore, it substantially reduced what's known as the regiodynamic sugar phosphate backbone once they ran the simulation. So they're refining their molecular modeling technique by using actual biochemical evidence, okay, which is what they need to do. That's what that paper was all about, by the way. So, and don't worry, all these papers will be put in the show notes. Let's go back to this. That was the 2012 paper. Let's go back to the 2017 paper. The overall structural similarity between formyl cytosine DNA and unmodified DNA does raise a question. What features of that formyl DNA do enzymes recognize? For example, that thymidine DNA glycosylase. You see? The results argue against the substantial shift in global conformation. That's what that data just told us. Even given the solvent effects, with the correction made the underestimation come back into register with what is normally found from actual experiments in solution. So in the complex with TDG, the DNA is severely distorted to form a structure in which the modified base is actually flipped out, okay, so that it is now external to the plane of the helix, and the flanking regions remain more B-like all the time than A-like. And that's basically how those two conformers of DNA are named in terms of the sugar pucker. Remember that slide a few minutes ago. So the overall reaction involves the enzyme binding to DNA, followed by, obviously, by then bending and inserting one of its amino acids as the enzyme is adding an amino acid residue from its sequence via the minor groove on the entire macromolecular structure of DNA to help flip out the desired base. Okay? That's what's going on here with this mechanism. That will then expose the glycosyl bond for the cleavage reaction within the active site now. So that whole substrate is moved over into the active site by this interaction between the protein and the DNA. See? And that has to do with water of hydration around the DNA and the hydrophobic effect as well. Now you get a structure in the absence of the enzyme. It would be a much higher energy, obviously, than the relaxed bDNA. And the population of flipped out bases would be very low in free formulated cytosine DNA. See? Free meaning without being bound to the polypeptide. That's what proteins do. They freeze the action of the reaction and then they catalyze. Right? They create a substrate in a higher energetic state that will easily go through the transition in the reaction so that all the energy was to build up that highly energized state so that the rest of the reaction is basically just all denouement. All the free energy has already just been dissipated out. So it's basically spontaneous. So logically flipped out base, a large number of interactions, sure, okay, so there's more surface area, and the enzyme can compensate for the local energy distortion of the duplex, which is what it does because it's bound, right? Now that S was supposed to be there, sorry. Specific interactions between the active site residues with 5-formyl or even the 5-carboxy cytosine would be sufficient for recognition for the enzyme, compared with the 5H, that's the hydrogen atom of the unmodified cytosine, because normally it's just a hydrogen atom there. See? Hence, the nucleobase cleavage rate would depend inversely on the strength of the bond. Obviously, again, which is lower, the bond strength is lower in the formulated cytosine and in the carboxy cytosine than for just the hydrogen atom. That would be the unmodified base. So formal cytosine is a conformationally restrained formal group because that aldehyde function. And the large dipole moment, of course, because of that as well, and the solution chemistry. So it can form a strong polar interaction with the enzyme recognition site. So the whole biochemical reaction now is explained very well between substrate and product, and how the enzyme mechanism alters the substrate to carry out the catalysis, right? So it's a spontaneous free energy cascade once that substrate is created in that groove in the enzyme complex. There's also an N1, that's a nitrogen atom 1 acidity, and a weaker nitrogen 1 carbon 1 prime, that's the nucleotide, that's the sugar. So under appropriate conditions, it's a good leaving group. Okay, so the chemistry is enhanced by this interaction. That's all this is saying. So it satisfies the binding and excision requirements of the TDG. The 5-carboxyl cytosine can also form strong binding interactions with that enzyme, right? But it is a poorer leaving group. Why? Due to the negative charge of the carboxylate anions. It's not a formyl group, it's carboxylate anions. So there's an energy charge distribution there. So the overall rate of reaction is going to be a lot lower than for the formulated cytosine. This is interesting, right? RNA polymerase 2 hydrogen bonds directly with the 5-carboxyl group. thereby reducing transcriptional efficiency. So this is another part about that whole thing, because you have that a carboxylate anion when you're talking about transcription, not TDG activity, but transcription, tRNA polymerase. You're going to get a reduced transcriptional efficiency because of the carboxylate anion. You see how this works? So endnotes. Although the control duplex and its 4-male cytosine containing counterparts, similar structures in solution. We've already covered that. That's the B form. Remember, we were talking about that. And they exhibit drastically different circular dichroism spectra, which can be attributed to the altered electronic transitions in the modified bases. The effect is not exclusive, though, to the 4-male CPG steps. That means the base stacking, right? Because in all sequence contexts that they examined, the circular dichroism spectra of DNA duplexes containing multiple formulated cytosine bases are strongly altered. That gives different spectra depending on the local environment of the 4-male cytosine. Okay, so we call that a relational category, a relation of where that formal group is, right? Relative to, you see. Don't worry, I'm going to get into this in more detail soon. The profound effects on CD then probably results on many local factors in that protein DNA complex. This is consistent with the abnormalities found in the absorption spectra using just UV. Finally, 5-4-male cytosine, that oxidation product of the methyl cytosine, doesn't change the global conformation of DNA, which was the original assumption in the modeling paper that it did, right? But rather, it operates at base pair resolution for the recognition of that site by the enzyme. Despite the small differences between formal cytosine-containing duplexes and their unmodified analog, it remains still possible that the formulated cytosine could alter mechanical properties of the DNA. I would say physical-chemical properties, perhaps by enhancing the flexibility of DNA, thus lowering the energetic penalty in forming a certain, that is TDG DNA protein complex, which would involve the bending of the DNA as a macromolecule. TDG could carry out that function. Right? That enzyme could. Now, let's do dialectic. So what are the categories that we've been talking about here? This is an aspect of epistemology and metaphysics that we're mapping onto the physical biochemistry we just went through. We're doing this so that we can always be sure to be within the rails of a track, following all the laws of thermodynamics we believe must be followed in what we're studying in living systems. Okay? So what are the categories of phenomenological events we addressed here? We have discussed modality, quality, and quantity. What's the modal phenomenological category involvement? This is what we observe in torsion angle redistribution versus this must be confined to allowed free energy states. Okay? So what we observe must be associated with what is allowed in terms of free energy. That was the conundrum with the early modeling study. What about the Quale, right? The quality phenomenological category, Quale. The arginine residues that we talked about earlier, not today's lecture, but one yesterday, will interact with the water of solvation proximal to the DNA helical major groove, which was caused by the hydrophobic effect, sorry for that misspelling, induced, another misspelling, by the methyl group. Okay? That's the Quale, the specific arginine residues in association with the nucleotides. What about the quanta? Okay, this is the quantity phenomenological category. A specific molar concentration of the MBD, remember that's the binding domain, the methyl binding domain of the polypeptide, and the DNA, that ratio, molar concentrations, obtains maximum velocity of the reaction. Now, relation is the final one. And we hinted at it 2 slides ago. It is the sequence that is the relation to the adjacent sequence, that's the step, that is specific to the epsilon zeta torsion angle redistribution. That was the results of that 2017 paper. So this isn't about how one sequence belongs to a larger sequence, that small nucleotide repeat within the entire nucleotide sequence of that region of that gene. No, it's not. It doesn't, because it's a small single series of sequence in a much larger sequence, but rather it is the event dynamics, and that's the relation itself. So it's not about the fact that you're looking at sequences, the fact that there is already a relation between the sequences. And that's what counts here in terms of the phenomenological event. So that obtains when the solvent dynamics are added in to obtain the appropriate free energy states that we know must be occurring for the relation to occur as an event in space-time. Now, if that's not clear enough, I think I gave you another yeah. So let's do a thesis, antithesis, synthesis to do a dialectic in the modern form. Phenomenological categories are okay, here's the thesis. Phenomenological categories are superimposed upon thermodynamic foundations of biochemical events. That's a thesis. That's a statement. Here's an antithetical statement to that. Not that. Thermodynamic restrictions obtain biochemical events through a phenomenological lens. That lens is provided by our senses, right? The instruments we use. Now let's do a synthesis of those two, because we have similar nomenclature in the thesis and antithesis. We can come up with a synthesis. It's not a paradox. Here's my synthesis. Empirical and rational mentation, which is what went through all this series of experiments we just talked about, are a priori to phenomenological categorization of the thermodynamic limits imposed upon the biochemical events. So the synthesis is the way that our mind works is a priori to creating the pattern of event ontologies so that we can understand the phenomena, which is basically the read in from our senses using our instruments. And that phenomenological characterization I just went through, right? Quantity, quality, modality, relation, then informs the thermodynamic limits that we say we're measuring, which we must impose upon the biochemical events we also measure and we observe. Okay? So it's a top-down synthesis, not a bottom-up. So it's not like all of these thermodynamic limits are just there, and then we build up those thermodynamic limits, right? And we get to an understanding of DNA solution chemistry when it's bound to a protein that's interacting with a methyl group on a cytosine residue, or a formyl group, which we spent more time on. No. It's a top-down. It's the fact that there is empirical and rational mentation that are a priori. It's how the mind works. Okay. All right. I'm going to stop there because the next thing to talk about now is to put this together in free energy coefficients. Okay? And that's what we're going to do next in authentic biochemistry. So let me find here how to escape this, end the show. Let me go back to here, to this slide, and we're going to say, from Authentic Biochemistry Podcast Studios, Dr. Daniel Guerra, bye for now.